First step of replication is when the enzyme helicase separates the parental DNA helix into two strands. This breaks the hydrogen bonds.
The next enzyme breaks, twists, and rejoins the DNA to relieve the tension caused by the separation of DNA. The enzyme is called topoisomerase.
Replication starts at the origin of replication, which is a short stretch of DNA having specific sequence of nucleotides. When helicase separates the two strands, it makes a replication bubble.
The replication of DNA proceeds in both directions. The replication fork is at the end of the bubbles, which would be right here. This is where the parental strands of DNA are being unwound. When the helicase unwinds, the unwound templates want to reunite, but single-strand binding proteins attach to each strand to keep them separated. Just like these magnets, if you had two positive magnets, they would push each other away, and that's just what the single-strand binding proteins do with the two strands.
Primase is an enzyme that composes short RNA strands called primers. Primers attach to a 5' end to make an available 3' end for DNA to attach to. They are needed so DNA polymerase can add DNA strands later on.
So this would be something to represent a primase, like a factory that's making primer, like this. So now there is an available 3' end for DNA to attach. This would also happen on the leading strand as well. As soon as the primase creates the RNA primers for the new DNA to attach to, DNA polymerase III will add new DNA nucleotides to the free 3' end of RNA.
So these are the DNA pol 3s. They create DNA on the available 3' prime end of the RNA on both the leading and the lagging strand.
In a DNA helix that's replicating, there's always a leading strand and a lagging strand. The leading strand is built continuously from the parent's 3' end all the way to the replication fork. The new strand is built in the 5' to 3' direction, so it's anti-parallel with the parental strand. So in this case, the leading strand is at the top of the DNA helix that was separated, and the DNA will continue from the primer right here, attached to the end of it, and continually make DNA replications towards the replication fork, like that.
The lagging strand on this DNA is on the bottom. The parental strand on the lagging strand is in the 5' to the 3' direction. Therefore, in order for the new strand to replicate, they would have to start at the other end and synthesize in fragments in the 5' to 3' direction. So it would continue down here until it reaches just before the 5' end of that primer. And this fragment is called an Okazaki fragment. After DNA nucleotides are added into the new strands, there are still the RNA primers left on the new strands. DNA pol 1 comes along to take out the RNA primers and replace it with the DNA nucleotide to complete the new DNA strand.
So it takes out the RNA primers and you're left with DNA.
But there are also gaps in between the new DNA here and the old DNA here.
So after the DNA pol 1 takes out and replaces the RNA primers, the ocosaic fragments on the lagging strand aren't connected fully. So DNA ligase comes along and connects the ocosaic fragments and completes the backbone.
Basically, these are finally connected, so they're just one long strand.
So while the whole DNA replication is going on, enzymes are always proofreading. So when base pairs are attaching, if an error occurs, the enzymes will remove and replace the base pair right away.
There's an enzyme called nuclease. It is the main enzyme that takes out the DNA bases that aren't supposed to be in that spot.
So for example, if adenine tried to connect with cytosine, nuclease would come along and take it out and replace it with the right base pair. Telomeres are non-coding nucleotides that are added to the end of newly made chromosomes to elongate the new strands so that the genes don't erode.
This is a double helix. This carries the genetic information in the body. There are two backbones of phosphate and sugars joined by base pairs.
The structure of the DNA helix is made up of a sugar, a base, and a phosphate. The nucleotides are joined by covalent bonds between the sugar of one nucleotide and the phosphate of the next. These are the base pairs here. Adenine and thymine go together, and guanine and cytosine go together. So the base pairs on the DNA helix would look like this under a microscope. And they bond together on the DNA helix like this. So adenine would go with thymine, and guanine would go with cytosine, like so.
T and A can pair with each other, and so can C and G. T and C are single ring structures called pyrimidines. A and G are double ring structures called purines.
A and T are complementary because there is one purine and one pyrimidine. They create double hydrogen bonds. C and G bond similar, but they have 3 hydrogen bonds. Nucleotides must be complementary in order for the two strands to attach and form a DNA helix.
Each DNA strand has a 3' and a 5' end. Therefore, the complementary strand would line up so that the 3' and 5' end will be opposite to each other.
That means that the strands would be anti-parallel. So, like I have on here, the first strand has a 3' end here and a 5' end here, And then the other strand is anti-parallel with the 5' being the opposite to the 3', and the 3' being opposite to the 5'. So this is the semi-conservative replication. It's the type of replication that DNA goes through. So it starts with one parental DNA strand, or helix, in the red, and it splits completely into the 2 strands.
The 2 parental strands act as a template for synthesis of a new complementary strand.